And it seems now we're at 8 o'clock, so I will start properly with an introduction and then I'll hand over to you, Roberto, okay? So, welcome to the International Hibernation Society seminar series. My name is Shona Wood. I'm a member of the International Hibernation Society board, and it's my pleasure to introduce Dr. Roberto Nespolo from the Austral University of Chile. Roberto is an ecophysiologist who's working on molecular and physiological adaptations to climate change in hibernators. And today he's going to tell us about a South American hibernator. So I'm just going to hand straight over to you, Roberto. Thank you.
Okay, thank you, Shona. Thank you, everyone, for coming. It's an honor to be here. I'm very grateful to have invited to this seminar to talk a little bit about our model organisms, which is this animal that you can see here. I am in the southern tip of America, in southern Chile. So one important thing to keep in mind is the temperate rainforest, which is an endemic ecosystem that is only found here. That marsupials, which is the animal I study, lives only in America and Australia. This is something that I'm gonna repeat again in a few minutes. But importantly, this is the place where Valdivia is. I work in the Australian University of Chile, which is this campus here. My laboratory is about here. About here. Right, so it's a nice city if you want to come with very good beer and those nasty animals in the coast of the river that tourists like so much. Those are big fur sea lions walking in the streets in Valdivia. So I work with a team of different student postdoc colleagues that you can see here. I'm not gonna go into details of everyone. So we have undergrad student, grad student working in different aspects of ecology, physiological ecology, evolution and genetics of hibernation and other aspects of these populations. We usually do some outreach experiences. I'm gonna show a little bit about this later, but the most important thing is that we are focused in energy. We study how the important thing is how energy goes through ecosystem and is packaged, right, in animals. So we are interested in scaling, in metabolic rates, and particularly in those kind of old and important conceptual bodies, such as the interspecific scaling, the Kleiber law of scalings in animals, that says that in general, small animals lost a lot of heat. So if you are in a small size, you will be in trouble in cold ecosystem, just like in this representation of the sphere and the smile sources related with the increase in metabolic rates, right? Because you need to compensate because you lose a lot of heat when you are very small. Then animals adopt different postures. The sphere is the form that least amount of heat. So if you are small like this, you're in trouble in an ecosystem like this one. Then we are interested in how these ecosystems are adapted to the cold, especially in high altitude. We are large animal with coats and cars, so we can go there with not so problem, and trapping small animals, such as here in the summer, sometimes digging for sample below trees, meter of snows and minus 30 degrees in Los Andes. Sometimes taking samples of microbes from trees with the main question of animal how facing the cold. So one of the things we have been doing, my friend and colleague Francisco Bubillo and I were intended to grow those lenga trees, the main tree species at the high altitudes, finding new species of yeast and also another that is the ancestors of the lager yeast, beer production worldwide, published it a few years ago, but this talk is about another kind of cold adaptation. This is just to show that we can do a range of things from microbes to mammals. And when we talk of mammals, usually at the south and hibernating mammals is this model called the Monito del Monte. This is a small marsupial that hibernates. We can be used for applications for a number of problems such as organ transplant, and I'll tell you later. The Monito del Monte is the only hibernation of South America demonstrated by 5 field studies of hibernation. Hibernation is the seasonal metabolic depression that many animals experience without loss of temperature control that was discovered at least 100 years ago in rodents of Northern Hemisphere, such as these dormouse described in the 19th century, a phenomenon that soon was identified in different kinds of animals, such as marsupials, carnivores, such as deer, many rodent, bats, and even in the echidna. So it's a polyphyletic trait that appeared, an adaptation that appeared differently in different lineages at the same time, independently. So in terms of the phenology, it looks like reduction in body temperature to environmental levels with periodic arousal, like this typical example of torpor boats in a large carnivore. Those torpor events in hibernation have duration of several days or weeks. So studying torpor and hibernation, we have to apply mechanistic explanation, for instance, to address torpor induction mechanism for gene expression pattern of cryoprotective protein. We have the evolutionary explanation as well, which includes phylogenetic comparative analysis to search for the origin and evolution of hibernation, of the hibernation phenotype. And of course, we have the ecological explanation aimed at knowing survival and bioenergetic natural population of hibernators, which is now our main interest. So let's see some geographical pattern. Hibernating species are mostly distributed at high latitude, where you have a map. See, if you have a map of those whole Arctic hibernation, they look like this. There are a smaller number of non-whole Arctic hibernation at the south that I put there, but very, very few of them have been studied using field approximation and combining to ecological analysis. Note that whole Arctic hibernation are northward distributed as are continents. So compared with non-whole Arctic species, that they are closer to the equator. So that's why if you compare the typical pattern of hibernation in whole Arctic hibernator, this is more or less like this one in the 13-line ground squirrel, and differed a lot from non-whole Arctic hibernators, such as this species, the Monito del Monte. This is some data that we published from years ago. So you can see torpor event in summer as well, our long torpor event in winter, which is defined as hibernation. So if we have been working with this interesting model of South American hibernator that is widely distributed. That's probably 1,000 kilometers of distribution in Chile and Argentina. Here you have Chile, and this is Argentina. And then you can see there are some high altitudinal population of the Monito del Monte, where they hibernate within holes on those trees. This is an arboreal animal. Some of them, so we were interested in a few years ago in this species in high altitudinal population, and especially interested in estimating how much energy they save thanks to hibernation. So, because really there are many ecological questions that depend on such calculation. Indeed, calculating energy savings of hibernation is not simple. Here you have an example showing that if you compare the predicted daily energy expenditure in hibernation in many species, you know that the savings are the highest in very small animals, such as monitos, of those very small bats of 9 grams of Canada and the Arctic. So importantly, if you save energy from a hibernation and you need to save this energy every year in these long torpor events, of course, winter increases of temperature will somehow affect this survival. And this is something that we were modeling a few years ago. So if winter temperatures go up a few degrees when animals are hibernating and obligated to wake up, so then if you are spending very small amount of energy in winter, a sudden arousal because of warming will make you need a lot more food to survive in a moment when it's not available. So the thing is that monitors could save 96% of the energy when hibernating. This is something that we measured 3 years ago. So then you have a savings of 4%, a reduction of 4% to 4% of the thermic level. This is probably one of the 3 or 4 more extreme cases of hibernation worldwide, reaching sub-freezing body temperature in the tissues. So you have very energetic animal, a marsupial with high aerobic capacity in summer that goes to very low metabolic rate in summer during hibernation. So we were interested as well in how these different levels, this large variation in aerobic capacity is explained at the level of tissues and cells. So this, relating to this prediction of survival to global warming, we were analyzing in simple terms, doing a physiological model to predict survival to global warming, estimating the threshold when animals arouse from torpor in winter, which has an equivalence in days of survival to winter. And then you can plot this model, this predicted model using a raster into the future scenarios for warming, for which we parameterized a model of energy budget that looks like this. But this is the important part. You have a different curve of energy consumption when animals are active or when an animal is in torpor. So we just modeled the probability to shift from this curve to this another curve with different temperatures. So for this, we analyzed it. This is published last year. So we to know the parameters for those models, we have been working in San Martin field station, which is crucial. It's very important to have a experimental station in the field to analyze and to capture and recapture and to measure things in those animals. This is a property of the university with the biggest monitor population recorded to date. We have 300 hectares with a relict forest with about 35 individuals per hectares, which we have been sampling monthly since 3 years until now. So we have 300 traps permanently installed there in the trees, because this is an arboreal animal. And we have some observation and research areas where we installed those cylindrical cages for putting the animals to be followed during the winter, doing a number of experiments. This is the map of San Martin Station, and this is an aerial photo showing our laboratory over there and different tracks. So all of these are native forests without intervention during the last 40 years. And this is for 1 hour far from our campus. So we have 5 grids. Every one of those grids have 51 traps, tomahawk traps for leaf capture, and there is the laboratory with those enclosures. So we have been following those population ecological. This is the thesis of Isidora, master thesis, and you can see that's important because you can see that the captures and the densities of the animal are actually increased in the last 3 seasons. So that's very important to justify doing research in molecular biology, on physiological ecology in such an important and unique animal. This is important to get the permit. So those are the enclosure we have. This is we should for calibrating this model I told you of survival to warming. These were the prediction here for calibrating the animal, the model, sorry. We have been doing these things such as manipulating food offered to the animals in those enclosures, right? This is for individual hibernating in those enclosures with data logger to measure different internal temperature using heat distribution hibernating group with thermographies, or measuring isotopically metabolic rate with double lavender water, which is something that we did in 2020, or recording the amount of fat and lean tissue by quantitative magnetic resonance, a very, very nice machines that can give you the amount of fat and lean tissue of an animal, a live animal, in a few seconds. So this permitted to measure fat and lean mass consumption in hibernation, as this graph at the left, that permitted to calculate the energy consumption of hibernation in those animals in semi-natural conditions. This is a very small number compared to other hibernators. So we have been published this and putting in a comparative allometric equation to see how much energy is saved by hibernation, but especially to build the model which gave this prediction. This says that an optimum hibernation temperature for this species is around 2 to 3 degrees Celsius, and survival to winter will be reduced 50% with only 2 degrees in increase of winter temperature, as is shown over there in this predictive map. Of course, this depends on the amount of fat the animal accumulated during autumn. So the projections, if you use the different scenarios for global warming in South America, looks like this for the next 50 years, extinction in northern Chile and survival in Argentina. Both here is at present. You can see that population of monitor are mainly and are dense, covering a geographic range of about 1,000 kilometers. But let me spend a little bit explaining why monitor are important after showing a little bit of what we have been doing in terms of global warming. Those Monito del Monte, two species, Dromicio bliroides and Dromicio bocinovici, are marsupials, are microveteriate marsupials, the only the only living representative of an order of mammal that went extinct 20 million years ago. So in addition, these animals, these monitos or these marsupials are more related to Australian marsupials, are considered a living fossil because of this condition, of this status, and are the known South American hibernator, and also are a key part of the ecosystem, as is the unique dispersal of several endemic plants in the temperate rainforest. The temperate rainforest looks like this. This is the range of distribution of the two species. This is like a tropical forest, but humid and cold. So we have in winter about 5 degrees in average, never freezing in the coast, but yes, below freezing near the Andean range. These are the different characteristics of the ecology of this animal. Are an animal and species with many roles. Then, of course, this is a marsupial, so the whole cycle is very important for during the lactating phase, because they don't have a placenta. So the whole cycle need to be covered in through to 3 months in the summer. Many places in Chile think and have an important iconography of this species. We have different paintings in Santiago or Valparaiso, or in Argentina as well. You have some beers named after the Monito del Monte. So it's a popular animal and is emblematic for science as well. But let me tell you a little bit about the origin of marsupials. Mammals are originated in the Permian over 200 million years ago. Here's at the right, I put an evolutionary cartoon recalling that mammals are the three groups of animals derived from therapsids, a kind of vertebrates that lived 200 million of years ago. Here we have a continental position, which back in the Permian were all together in Pangea. So then Asian fossils revealed that the origin of marsupial in China, but we don't have marsupials in Asia right now. And then they dispersed to North America in the Cretaceous. And somehow they managed to get South America, where microbacterial marsupial diversified. So those microbacterians passed through Australia by an Antarctic bridge, while these land masses were still born 65 million of years ago. Then later Australia became isolated, and in South America, microbacteria went extinct, excepting for dromiceps, the Monito del Monte, that took refuge in southern South America in those temperate rainforest. So this story was confirmed a few years ago with the first sequencing of dromiceps genome we did in collaboration with some colleague in China. So the Alta marsupials diversity came from Asia, dispersed to Australia through America, and America retained the ancestral undifferentiated opossum-like form with three groups. But Australia experienced the great diversification of marsupial form in all the ecological niche we see for placental in other places. But having in the middle the Monito, Dromiceps glirioides, an ancestral living form of Australopithecus marsupial. Now we know these are two species of Monitos, Dromiceps oceanovici at the northern range and Dromiceps glirioides at the southern range, with an ancient history of divergence dating 13 million of years ago. As we calibrated the genomics with fossils, that was the PhD of Julian Quintero, and was published over there. Interestingly, this calibration is coinciding with the fossil record for microbacteria. So Dromiceps in the Cretaceous, Dromiceps is identical to those fossils named Microbiotherium, a fossil species that got extinct in the Miocene, in the middle Miocene. So this is a survived lineage that as well is the only known hibernator in South America. But let's go back to marsupials model. Here we have an albine monodelphi domestica obtained by gene editing. By far, this is the most used marsupial in biomedicine. Some other species have been also useful, such as the large one of the kangaroos in Australia, particularly this amazing embryonic diapause they experience or the great resistance to pathogen that the Delphis virginania exhibits. So we have been playing seriously with the idea of using hibernation physiology to try to save human lives by somehow inducing metabolic suppression in ex situ experiment in pig livers, because this is known that hibernation protect their brain by overexpressing antioxidant enzymes, neuroprotective peptides, and then right retraction for protection, which was also shown in the Monito a few years ago in collaboration with Ken Storey. We did this transcriptome study in which we detected several differentially regulated genes in the brain, preserving organ function, inhibiting angiogenesis, and prolonging life by inhibiting apoptotic pathways, among other things. So we collaborated in this with Ken. Sorry to say, Ken passed away this year. He was a tremendous collaborator and very generous and teacher in hibernation in other aspects of physiology. So with other author, we have found that hibernating organs such as muscle reduce oxidative damage and avoid atrophy even after months of inactivity. I have some stickers, some rare stickers of monitor, hibernating monitor, especially designed for this slide. It is clear that hibernating species live longer and suffer less oxidative damage, something that we could like to explore in the Chilean monitors. So my plan is to use a new grant we just submitted to apply to study organ preservation using hibernation cocktail, for which we have a colleague, a medical doctor in a laboratory using porcine livers, the team of Rolando Rebolledo at the Institute for Translation Medicine in Santiago, who work with an ex situ system for organ maintenance. Indeed, another plan we have is to develop a primary cell culture, which is actually worked a few months ago for liver and spleen and kidneys, and to see if we can induce them with immortality and perhaps in the future have organoids of hibernator, which is of course long-term ideas for next steps. This is something that we are doing in collaboration with Ignacio Mieci at the Biochemical Department of Universidad Austral de Chile. But we also have a field laboratory that was set up a few years ago. You see a small laboratory with a respirometer for doing bioenergetics in the field. Here we are usually measuring maximum metabolic rate, basal metabolic rate, some biochemistry and blood parameters for analyzing oxidative damage. So here you have the animal. This is a male monitor in the middle of breeding. They develop those large testes when they're breeding in the breeding season starting in November, December. And here you have a monitor doing some workout for measuring maximum metabolic rate, which is the maximum capacity for sustaining work, which is this is a metabolic chamber. So we record when the animal get to the limits, and with this kind of studies combined with some behavioral analysis that we are doing right now. This is an arena during the night, and this is a monitor for which we are measuring the time they explore, they climb this streak, and related with this velocity and exploration time with maximum metabolic rate and aerobic capacity, we have been reaching this kind of result. This is unpublished, showing that maximum metabolic rate is correlated during the summer season with the exploration parameter, as well as this other kind of data. This is just published this year, showing that aerobic capacity is duplicated during summer and have a reduction during winter. So this difference is about 2.5 times in summer compared to pre-hibernation in winter, which is kind of a record of training, because human, for instance, cannot obtain 40 or 60% increase of aerobic capacity by training, for instance. Here you have the animal. You have differences in summer animal and same animals in winter. We have been using some nest cages. We can those are social foragers. So you can see all of the animal fighting for the food, and then you can see in one of the nest cages another animal absolutely very active in summer and absolutely very metabolically depressed during hibernation in winter. So we think it's a very interesting model for analyzing different aspects of this capacity of changing aerobic capacity. For instance, we have those preliminary images of the muscles. These are muscles of monitos. You can see the large sarcomeres that are usually quite more large than human sarcomeres. And this is the comparison of humans before and during space travel, showing the reduction in the size of the muscle fibers. And especially one of the things that was very interesting for us are those huge mitochondria located within the muscles. So this is something that we are developing now, comparing animals from the metabolic depression in winter with the very active and trained animal in summer from field studies. So this is the trapping grid, and in general, this is what we are applying in our new grant. This is a summary of the differences of hibernating animal in winter and the metabolic expression in summer with the differences at the level of tissues, lipid-based metabolism, reduced peripheral irrigation, repression of expression of many metabolic pathways compared to the carbohydrate metabolism in summer, very active, very moving in the canopy of the forest with a lot of production of reactive oxygen species. So we plan to train animal in this typical system of wheels and analyzing muscles in the new grant that will be in the next 4 years if I get the grant. And this is the summary of the general design of these experiments we will be doing hopefully next year. So this is my general summary of our work. I would like to receive any questions, any discussion. We plan to, in the long term, to apply those hibernation physiology to organ transplants as on preservation, as I said. And some people believe that this could be applied for space travel for inducing hibernation to humans. So this is, we have some monitors over there saying goodbye, and this is how they think. This is the first real sound of an animal breeding, a male breeding in spring. They produce this sound to attract the females. So thank you very much for your attention. I will be happy to receive any question or comments.
Thank you very much, Roberto. It's very nice that you gave such a comprehensive talk, showing us how it's very possible to be able to encompass ecology, field studies, physiology, and molecular biology all in one program. Very impressive. Thank you.
Thank you.
So I'm going to open the floor to questions. If you raise your hand and then I will then unmute yourself and ask your question, that would be very good. Thank you very much. So we're just looking to see if we have anyone. Perhaps I'll start the questions and then other people can pitch in. So I was wondering if you've done any studies where you bring the animals into the lab and manipulate photoperiod to induce hibernation. Is that possible in these hibernators?
Yeah, we did this many times. It's very easy. We can bring the animal from the field to either to the laboratory at the field, or we can bring them to the laboratory at the university. We have seen that photoperiod is not the most important induction for torpor and hibernation. The most important thing is cold, but by far the most important thing is the lowering glucose in the blood. So you just remove the food and they go torpid after 1 or 2 days. So if the environment is cold, that would be longer, the torpor period. That usually
Okay.
the same in all hibernators.
So is the depth of the hibernation similar between the food restricted and the seasonally prepared hibernation? Is it?
Yeah, we did an experiment. We published a paper in 2022. We did an experiment in the field where we removed completely the food, and there was another few enclosure with food ad libitum, and they started to do torpor as usual, but then the animal with increasing food aborted hibernation. So if you give food them, they maintain they are maintained active.
Okay. Yeah. Thank you. It's nice to know. Does anyone have any questions? Open the floor to other participants. Stam, yeah, go ahead.
During the winter hibernation, during the arousal from low body temperature, do they emerge out of the burrows at all? Or, and if they do emerge, is that related to the available energy for each animal? Do you know?
They usually they experience periodic requirements during winter. So every 1 week, or even the maximum is about 2 weeks of torpor, they need to rewarm and stay euthermic for a few hours. They usually don't eat food in those periods. They just sleep and stay in this place. And this is believed, this is needed because they need to rewarm to eliminate toxic compounds from the blood, and they actually need to rewarm to get some sleep, because they are in torpor. The animal is completely hypothermic. The brain activity is very low, so they don't sleep.
But they don't emerge from the burrow, though. They stay in the burrow.
Oh, right. They stay up in the trees. They hibernate in the trees in burrows 20 meters above the ground, but they don't emerge. There's some time we have discovered some juveniles going out in the middle of winter, just seeking for some food, and we believe they are too small to maintain longer hibernation. The mortality of the juveniles is very high. It's about 80%. So once they get the second year, 30 grams of size, more or less, they start to do the normal cycle of hibernation.
Thank you.
Okay.
Jessica, go ahead.
This is really interesting. Thank you. I had a question about sex differences, and it looked like there were at least sex differences in recapture rates and their kind of activity patterns. But I'm also curious about what that looks like in terms of their energy expenditure and how they're balancing that with their torpor.
Yeah. Morphologically, there are almost no sex differences, other than the sexual organs. But in behavior, we have seen that males start first. They emerge first in October, and they cover all the canopy. They have 3 or 4 times more movement than females, and they start to appear in November. So we think they have some breeding system of polygenic breeding system of many males copulating with one female. That's why we saw in this time series a lot of more male recapture, especially in the third year, 2 or 3 times more male recaptures in spring, some female, then this get equalized, get the same in summer and after breeding. So that the sex ratio is so different in spring.
And do you see any differences in their energetic?
I haven't seen. Yeah, we have measured many times different kind of metabolic rates. This mostly the same. It's the male don't have more energy expenditures than females.
Thank you.
Okay, thank you. Does anyone else have a question? And if I haven't noticed you waving your hand, just unmute your microphone and ask the question, just in case I didn't see you. Anyone else? So perhaps I can ask another question while other people are thinking. So I may have missed it because it's not really my strength, the mathematical part of the work that you've done with the modeling. Did I understand that you meant if there was a 2.5 degree C increase, you would have a loss of survival, or was it that 2.5 degrees was the correct temperature for them to hibernate at? I wasn't quite sure.
What we found is that the winter temperature increased by almost 2 degrees. You will have a 50% more mortality. That's the prediction. So we put this in the map, every pixel of South America for the next 50 years with the different scenarios for global warming. And we found those areas of mortality and extinction and some areas of survival, especially to the south and Argentina.
So if temperatures increased even more, though, there must be a threshold where hibernation would no longer be beneficial and food supply would be higher, so therefore they would increase in survival. Is that part of your modeling?
That's the different. Well, there's a debate, of course.
Yeah.
In the northern hemisphere, some authors have proposed that, for instance, for the marmot and for some rodents that lives in the Arctic. So some authors have seen that with the increasing temperature, there will be more food, right? This will compensate the mortality during the winter. But we argue that first, you have a lot of oxidative damage when those animals that are used to evolutionarily adapted to hibernate stop hibernation. That's what happened when we gave them food, for instance. A lot of food, they remained active during winter, and there was mortality in the summer because they need to be for a number of days in hibernation. That's one thing. And the other thing is that the ecosystem of temperate rainforest is not that high in latitude. So it's a forest that is very stable. You won't get more food if temperature increase. Everything is very stable and depends of the flowering, which is always the same. So this is an animal that is frugivorous in summer. So I don't think this reasoning of those northern hemisphere hibernators applies so exactly for this southern hibernator. But it's something that is discussed right now. There's a paper in PNS of some French colleagues of 20 years of data set showing that the marmot are increasing their population. But this is a completely different approach than we used. One approach, just to finish the answer, we applied one approach that Humphreys and Spickman applied in a Nature paper in 2002 for the small myotic bats of Canada. We reproduced this model and reparameterized this model for this another hibernator to the south.
Thank you. It's very interesting to think about the contrast of how environmental shifts will be very different depending on your hemisphere. Yeah. Does anybody else have a question?
There was a question here.
Yes, Yoshifumi, go ahead. Yeah.
Hi. I'm Yoshifumi Yamaguchi from Hokkaido University in Japan. Thank you for a very nice talk. So I'm interested in the timing of entry into torpor. So even in summer, when food is restricted, this animal enter torpor. So is the timing of the entry dependent on the cytogeber or circadian rhythms?
It depends of temperature. Usually it depends of temperature and food availability. So that's why we have daily torpor in summer. Sometimes you can have here in the middle of summer, you can have one day with 5 degrees. You can find the animal in torpor for one day. So this is kind of random. Usually they don't go to torpor in summer and spring, but it can happen. This is a very different aspect with northern hibernator that is strictly don't go to torpor in summer, and they suddenly start to do torpor in autumn. And in autumn they start to do more extended period of torpor about April. Then they get in May completely hibernating. So it's mostly depending on temperature. We haven't seen photoperiod effects, or not clearly as in some northern hibernators, which is very clear. It is in the hamster, in the dormouse, in many different hibernators. This is a clear effect of reducing photoperiods. The work of Gerhard Heldmeyer 20 or 30 years ago with melatonin, showing clearly that there's something about light hours. I don't know if I answered the questions.
Oh, okay. Thank you. So I'm asking about the when the animal enter torpor in
When?
the day. When? Timing.
In the day, right? It is a nocturnal animal. It's active during the night. They start torpor when they do it when it's at the end of the night, in the morning. So the most deep torpor event and during the morning, and they sometimes start to wake up warm in afternoon. But if it's cold, they remain in torpor.
Okay, thank you very much.
Okay, we're getting close to the end of our time, but if there is another question, we can have it.
Excuse me, may I ask a question?
Yes, of course, Gen, go ahead.
So, my name is Gen from Japan. I'm slow and and I think you have talked about the difference between the northern hemisphere hibernators and southern hemisphere hibernators, and what causes the difference? I mean, are there the metabolic difference, or would you explain more?
The explanation is mostly on evolution. The northern hemisphere have if you see the globe, the northern hemisphere has a lot more land to do the selection and adaptation experiment, because torpor and hibernation is an adaptation mostly to save energy during the winter. So we have a lot more different kind of hibernators in this northern hemisphere because of the cold, basically. So if you go to the south, you get to South Africa, which is not very cold. Then you have South America with those environments like us with this forest, and then you get to Australia. So there's not many places to evolve this hibernation as an adaptation. There's a very nice paper published, I think last year, in functional ecology, showing exactly, this is a phylogenetic analysis, I can tell you, I don't remember the author, showing exactly what are the evolutionary triggers to explaining why Northern Hemisphere hibernator are more diverse, hibernation is more profound, and of course, body size is one of the most important explanator of deep hibernation, because small animals are more constrained during the cold in the winter. So that's why the championship of hibernation are those very small bats, Myotis lucifugus from the Arctic. And the other champion of hibernation is the Arctic ground squirrel, which is was studied by Brian Barnes and Toyen in the Arctic Studies Institute. This is a small, it's not small, but it's not that small, but this is a very cold environment, reaching -4 degrees Celsius in the abdomen. No difference in metabolic rates, but difference in the evolutionary triggers like body sizes and land masses to evolve
I see.
adaptation.
Thank you very much.
Thank you.
Okay, do we have any more questions? No? Well, I think with that, then we can thank Roberto for a very nice presentation and a very stimulating discussion. And thank you, everybody else, for attending.
Thank you for inviting me. I will be very happy if someone has another question or wants some of the paper, just write me an email. I will rapidly answer all of those kind of interesting questions if you want.
Excellent. That's really good. So thank you very much. And as usual, the seminar and the discussion will be available on our YouTube channel for the International Hibernation Society. So yeah, and if you're not a member of the International Hibernation Society, I encourage you to join. Okay, so thank you very much. And with that, I'll close the seminar.
Okay, goodbye.